Hello and good evening to my fellow classmates and lecturer. In this video, our group is going to talk about the immune system. Immune system consists of innate defense system and adaptive defense system. Innate defense system includes external physical and chemical barrier, which is first-line defense and second-line defense. Skin and mucous membrane of the body are the first-line defense against pathogens. This external barrier prevents pathogens and foreign substances from entering the body and causing disease. If pathogens manage to overcome the first-line defense, do not worry, because we still have second-line defense. The pathogens will encounter second-line defense upon entering the body. This second-line defense consists of phagocytes, natural killer cells, and antimicrobial protein. Another defense mechanism in our body is the adaptive defense system, which is the ability of the body to defend itself against invading agents by recognizing foreign substances and provoke immune system responses called antigens. The third-line defense in adaptive immunity are cell-mediated immunity and antibody-mediated immunity. It needs to be exposed to the antigens in order for the immunity system to react. The cell-mediated immunity is effective against viruses and bacteria inside the cells, while antibody-mediated immunity works mainly against viruses and bacteria or fungi that are in body fluid outside cells. Antigens are the substances that can trigger adaptive defense of the body. The term antigens is derived from its function as an antibody generator. They act as ultimate target of adaptive immune responses. Antigens are introduced or non-self. Most antigens are large and complex molecules that are not normally present in the body. There are two important characteristics of antigens, which are immunogenicity and reactivity. Immunogenicity is the ability to trigger immune response by stimulating production of specific antibody, while reactivity is the ability to react specifically to antibodies or cells it provokes. Strictly speaking, immunologists define antigens as substances that have reactivity. Substances that have both immunogenicity and reactivity are considered complete antigen, but if the substances only have reactivity but no immunogenicity, it is considered as incomplete antigen. Typically, only a small part of large antigen molecule act as triggers for immune responses upon binding to antibody. These parts are called epitopes or antigenic determinants.
Major histocompatibility complex antigen. So what is MHC? MHC genes is a group of genes that code for proteins found on the surfaces of cells that help the immune system recognize foreign substances. This protein is known as major histocompatibility complex antigen. In human, this system is known as HLA system. Properties of MHC. First, the MHC is polygenic. It contains several different MHC genes so that every individual possesses a set of MHC molecules with different ranges of peptide binding specificities. Second, the MHC is highly polymorphic. That is, there are multiple variants of each gene within the population as a whole. These two separate properties of the MHC makes it difficult for pathogens to evade immune responses. MHC antigen play a pivotal role in the adaptive branch of the immune system. MHC presents peptides on the cell surface for recognition by T cells. There are three types of MHC: MHC1, MHC2, and MHC3. MHC1 molecules are expressed on almost every nucleated cell in the body. Hence, they present endogenous antigen that originate from the cytoplasm. However, these endogenous antigens can be either self-proteins or foreign proteins such as viral proteins produced within the cell. Upon presentation on the cell membrane, antigens are recognized by cytotoxic T cells. Let's talk about the structure. MHC1 is composed of three alpha domains, alpha 1, alpha 2, and alpha 3, and a single beta domain. Alpha domains are encoded by chromosome 6, while the beta domain is encoded by chromosome 11. The alpha 3 domain serves as the membrane-spanning domain. The alpha 1 and alpha 2 domains consist of most variable amino acid sequences, and antigens are bound to these two domains. MHC class 2. MHC2 refers to a class of MHC molecules mainly found on antigen-presenting cells, such as macrophages, dendritic cells, and B cells. MHC2 molecules present exogenous antigens. These exogenous antigens originate extracellularly from bacteria-like foreign particles. Antigens are recognized by helper T cells. These activated helper T cells release lymphokines, attracting other cells that destroy the antigenic material. Moving on to the structure. MHC class II molecule is composed of 2 alpha, alpha 1 and alpha 2, and 2 beta, beta 1 and beta 2 domains. Both alpha and beta domains of the MHC2 molecules are encoded by chromosome 6. The alpha 2 and beta 2 domains serve as the membrane's binding domain, while alpha 1 and beta 1 domain serve as antigen-presenting domain. The last type of MHC is MHC class III. It is a group of unrelated protein. MHC3 do not share any similarities to MHC1 and 2. It is completely different in function and structure. It participates in other aspects of the immune system. The MHC also contain a variety of genes that code for other proteins such as complement protein, cytokines, and enzymes that are called class III MHC molecule. Example, MHC3 is involved in complement system and the production of inflammatory molecules. Effects of MHC. Beta cells are activated to produce antibodies that eliminate or neutralize extracellular pathogens. Macrophages are activated to kill pathogens living in their intracellular vesicles. Bacteria or virus-infected cells are also killed to protect the other cells from further infection.
Okay, so you already acknowledge what exactly antigen is, its functions and characteristics as well. In order our immune system to respond, we need an antigen receptor to trigger the process. Generally, we can find antigen receptor at B and T lymphocytes. Firstly, let's talk about receptor on B lymphocytes, or commonly we call it as a B cell receptors. This receptor is basically an antibody that is not secreted, but it is anchored on the surface of B cell membrane. For your information, all antigen receptors found on a particular B cell are identical, but it is different on other B cells. So, each B cell can bind to only one type of antigen at one time. As you can see the structure here, each B cell receptor has 2 antigen binding sites. These binding sites will bind to the particular area of free antigen which termed as epitope. The binding occurs if their structures are complementary. If they are, epitope and receptor fit together like 2 pieces of a puzzle, which is necessary to activate B cell productions of antibodies to fight the disease. Next, T lymphocytes have T cell receptors. It is a protein dimer that embedded on the T cell surface membrane. This is the basic structure of a typical T cell receptor. Unlike the basic antibody molecule, it has only one antigen binding site. The receptors on T cell functions differently from those on B cells. T cell cannot bind to free protein antigens. Therefore, a molecule known as antigen-presenting cells, or APC, is needed to recognize and bind first on the antigen before the receptor on T cell able to bind with that particular antigen and thus continue to antibodies production. This table shows the comparison between B and T lymphocytes. Each lymphocytes have their own specific receptor that located on the cell membrane and they stimulate the productions of antibodies. However, they are different in structure and processes. Let's recap. B cell receptor has 2 regions for antigen to bind, while T cell receptor has only one binding site. Antigen presenting cell or APC are needed to initiate the binding of epitope to receptor in T cell, while B cell can freely bind to antigen.
An important factor of the immune system is the ability to differentiate itself from external materials. Since our bodies are continuously exposed to pathogens such as viruses, this amazing capability is crucial. The root of its ability to differentiate between both is known as presentation of antigens to immune cells. The body promotes an immune response to protect itself once an immune cell identifies its specific antigens. Antigens are presented on the cell exterior surface by two classes of proteins, HEMAC class 1 and HEMAC class 2. Now, let's get started with the HEMAC class 1 molecules. Proteasome is the place where the cytosolic proteins are transported. A proteasome is a complex which degrades proteins into small peptides by proteolysis. TAP transporters helps the peptides to transport into the endoplasmic reticulum. TAP interacts with the MAC class 1 molecule, which is associated to several helper proteins. This happens inside the endoplasmic reticulum. The term MAC stands for Major Histocompatibility Complex, found on the surfaces of cells that help the immune system to recognize foreign substances. The TAP transporters and helper proteins detach when the MAC class 1 molecules permanently binds to one of the peptide fragments. Now, the antigen-loaded MAC class 1 molecules is pinched off into a vesicles and travels through the Golgi apparatus to the cell surface. Discharge of the antigen MAC class 1 complex is a result from fusion of vesicles membranes with the plasma membrane. CD8 T cells now can recognize this complex by effector. Now, let's take a detailed look at the MHC class 2 antigen processing pathway. Endocytic activity leads to exogenous antigens to be taken up by the cells. Antigens interact with MAC class 2 molecules inside the endosome. Small molecule can block the binding site of these MAC class 2 molecules. Regardless of that, another helper protein present in the endosome is capable of facilitating the exchange of these small molecules with any available antigen. The vesicles then travels to the cell surface, fuses with the plasma membrane and presents the antigen MAC class 2 complex on the cell surface. Now, the CD4 T helper cells can recognize the antigens. So, what's the main purpose of these two antigen processing pathways? Antigens presented by MAC class 1 are referred as endogenous antigens and derived from the inside of the cell. These antigens can be originated from the cell, representing a normal self-antigen or neoantigens which are derived from cancer cells. Viral components from virus-infected cells are the other endogenous antigens. On the other hand, MAC class 2 antigens are referred as exogenous antigens and acquired from outside of the cell. Thanks for watching.